chemistry from UC Berkeley, where he worked with Dr. Pete Schultz on cyclin-dependent kinase inhibitor discovery before moving here to San Diego to work at GNF, where he was director of biological chemistry. During this time, Dr. Gray's team made discoveries that led to the development of numerous FDA-approved drugs, including anti-cancer kinase inhibitor ceritinib, ABL001, and the multiple sclerosis drug siponimod. Nathanael in 2006 moved to Dana-Farber Cancer Institute and Harvard Medical School, where his group has continued to work on kinase inhibitors, making early discoveries that led to the development of FDA-approved covalent EGFR inhibitors. Oh, sorry, they can't hear me. Hello? Okay. Well, in 2006, Nathanael moved to Dana-Farber and continued to work on kinase inhibitors, making early discoveries that led to the development of FDA-approved covalent inhibitor osimertinib, and developing a wide array of chemical tools for transcriptional kinases, immune kinases, orphan or understudied kinases, and LOG2, a favorite target of lots of people in this audience, many of which are being advanced into new medicines in the biotechnology sector. More recently, the Gray Labs focused on development of targeted protein degraders, which I think we're going to hear about today, against kinases, recalcitrant drug targets, as well as the development of new technologies such as chemical genetic degradation tag systems and novel proximity-based approaches. So all these accomplishments have been recognized through numerous awards, including the NSF Career Award, the Damien Runyon Innovator Award, American Association for Cancer Research Team Science and Outstanding Achievement Awards, and the Paul Marks Prize in 2019. So all of this you can find out online, but what it doesn't say is that the Gray Lab is actually a super vibrant, exciting, and collaborative place to train and do research. So very grateful for the opportunity to do my postdoc there. And Nathanael is a truly, truly incredible mentor. The Gray Lab recently moved to Stanford, so really looking forward to hearing the latest research from the group. Thank you so much for visiting us, and I'll hand it over to you.
Thank you, Fleur, for that really kind introduction, and thanks everybody for coming out in person. It's great to be back in San Diego and to be able to do something live. I lived in San Diego from '99 till 2006, and I worked up at the Novartis Genomics Institute. So being back on campus, it reminded me of the only time I've had a moving violation. I was pulled over by a policeman here while on my bicycle for doing a rolling stop sign. And then he almost wanted to cite me for evasion because I couldn't believe that a car was actually chasing me on my bicycle. So lots of fun memories. So these are my disclosures. Our lab gets quite a bit of funding from Deerfield, which is a VC group interested in protein degradation, and I was involved with a startup doing protein degradation called C4. So I thought what I would do today is get everybody up to speed on protein degradation as a field, where it's come from in terms of small molecule modulation, and then just go through some case studies of how we've explored this space, looking at BRD4 and various kinase inhibitors. Then I'll talk a little bit about this degradation tag system, which I think is probably the most useful system our lab has developed for degrading a target of interest when you don't have a small molecule. And then I'll try to end talking about these molecular glues and how we're trying to find molecules that glue ligases to new targets to induce their degradation. So I think all of you know the field of pharmacology has been dominated by classical occupancy based inhibitors, where typically you're finding a small molecule that binds in an enzyme active site or an allosteric inhibitor. The molecules I'm going to talk about today are different. So they sort of come in two flavors. One are these bivalent degraders, which bring two proteins together via a intentionally designed bivalent molecule, or the thalidomide sort of type compounds, where it's a single small molecule that brings together two protein surfaces. So this field really grew out of a very old and infamous drug called thalidomide, infamous for inducing really horrific birth defects when dosed inappropriately during third trimester of pregnancy, leading to its ban. And then amazingly, later it was serendipitously discovered to have very strong activity against myeloma tumors. And it wasn't until approximately the 2008 or 10 timeframe when Hiroshi Honda and others figured out that what these molecules do is they actually bind to a class of E3 proteins called Cerebellum, and upon binding, induce degradation of specific substrates. So a series of studies from Ben Ebert and from Phil Chamberlain, and then also from Bill Kalin, they found that basically when these molecules bind Cerebellum, they induce degradation of what are called neosubstrates. So instead of Cerebellum recognizing its normal client proteins, these compounds glue new proteins. And in the case of blood cancers, there's two transcription factors called IKZF1 and 3 that are degraded, and that causes these compounds to be able to kill myeloma cells. Parallel to this effort, there was a beautiful work happening from Craig Cruz and Ray Deshays, where they were essentially trying to design the same type of mechanism intentionally. And so what they were developing were these compounds that they had called PROTACs, which were going to intentionally ligate a target of interest with a ligase. Actually, the ligase that they had primarily worked on was another ligase called VHL, which normally recognizes hydroxylated proline substrates. And the problem in the field had been that these compounds would typically only work at about 10 to 20 micromolar concentrations, and everybody thought this was sort of limited to the realm of chemical biology. But once it was discovered that these IMiD class drugs, which were working extremely well clinically, worked by this mechanism, that really galvanized the field to think, okay, maybe this is going to be more general, and we're going to be able to make degraders for lots of new targets of interest. Of course, this is not a new concept in nature. There's many, many natural products, that work as, molecular glues. And I give some examples here. For example, rapamycin, which is an allosteric mTOR inhibitor, only blocks phosphorylation of certain mTOR substrates through recruitment of FKBP, or beautiful examples from the plant biology where you have abscisic acid acting as a dimerizer. It's actually much easier to find a small molecule dimerizer than it is to find a small molecule inhibitor of a protein-protein interaction, because you only need to add, a small amount of additional binding energy, often to get two proteins to associate with each other. So we got really excited by these discoveries, and we really wanted to try to learn, what are the rules and the right approaches for making these kind of degrader molecules. Because in theory, the excitement here is that you could target, proteins that you don't know their catalytic function, or they may not have a catalytic function, and you could eliminate them, and you might get better congruence with genetic approaches, that deplete protein levels through various RNAi or CRISPR-based approaches. So the first system that we started working on together with Jay Bradner's lab was BRD4. So BRD4 is a well-characterized transcriptional regulator. Its normal job is to bind to acetylated lysines on histones and help form these massive complexes that regulate gene transcription. And so Jay's lab had made the first example of a selective ligand for BRD4 called JQ1. And so initially, what the lab tried to do was turn this binder of the acetylated lysine domain into a degrader by linking it via a linker to thalidomide. And lo and behold, they got a very potent and selective degrader. So you can see here 100 nanomolar of this bivalent compound. And after 1 hour and 2 hours, BRD4 is completely eliminated from the cell. And I actually remember when Jay's lab got this result, he came running over and showing us this plot. I mean, it looked miraculous that you could just put on this tiny molecule and get the protein to disappear. So we started thinking about this, about what are the requirements for a degrader molecule versus a conventional inhibitor, right? So conventional inhibitors, you're taught in pharmacology class, about the need for residence time, the need for continual drug exposure, and typically that you're inhibiting some enzymatic activity. These degrader molecules are totally different. So now what you're thinking about is a molecule that induces transient association, and that your pharmacology is now being driven not by inhibition, but by elimination of that protein function. And finally, you have the opportunity that a single small molecule can catalyze the degradation of multiple protein turnover. In some sense, it's a very biomimetic strategy, because the way ubiquitination is normally regulated in cells is you've got these substrate adapters for the E3 ligases. There's about 800 substrate adapter proteins that recognize all sorts of obvious and sometimes totally unobvious degrons on proteins. And so essentially, we're making the chemical equivalent of substrate adapters, except they're small molecules. So they're like substrate adapter small molecules that can then induce de novo protein-protein interactions, leading to degradation. So when you start thinking about this as a biochemist and as a chemical biologist, you realize you need a whole suite of new assays to discover and to characterize these compounds, right? So you need the ability to measure binding to the target and to the ligase. These molecules are big, they don't obey Lipinski's rules, and oftentimes they're, a little bit too big, a little bit too polar to get through cell membranes. And so you really need to assess, when you're actually able to get into cells. You need good ways to assess target degradation, how long the target is degraded, and obviously you're only affecting the kinetics of degradation. Upon degradation, every protein has different kinetics for how quickly it comes back. And so if your protein comes back more slowly, then you're maybe more likely to see pharmacology from a degrader versus a protein that can get resynthesized extremely quickly. Then, of course, you need to understand which cell types you're degrading in. And you have to understand, how drug exposure relates to where you do or don't see degradation. And there's a very important lesson there from thalidomide, because thalidomide was thought to be a really safe drug because you can give rodents huge doses of thalidomide and you don't see teratogenicity. And now it's actually understood why there was that confusion. It turns out that there's a mutation, an amino acid difference between human and mouse cerebellum that causes mouse cerebellum not to degrade the key substrate related to the teratogenicity. And there's a beautiful series of papers from my colleague, Eric Fisher, showing another transcription factor called SAL4, which is neuronally expressed, seems to be the culprit behind the teratogenicity phenotype. And so you can totally fool yourself with a degrader molecule because there's a species difference in the protein-protein interface. And it can either be a species difference on the side of the target that you're trying to degrade, or a species difference on the side of the ligase. So that really means that when you do your preclinical safety pharmacology, you've got to try to match the right pair with the target. So one of the excitements in this area was that we hypothesized and then were able to show later that you really can get some unique features with these compounds, right? So why are antibody drugs typically more selective than small molecules? Well, they've got more molecular real estate to recognize their targets with. Here with the small molecule, you get the same phenomena. You get the small molecule protein recognition, but then you get that protein-protein interface recognition as well. So you basically combine the world of small molecules with the world of biotherapeutics, where you get these bigger protein-protein interfaces. And I'll show how that allows you to get some unique selectivity. The second point here is that with traditional inhibitors, if you get a mutation that reduces the affinity tenfold, that can then have a tenfold difference in your target occupancy. But because these molecules are catalytic, even if you have tenfold less occupancy, if the molecule is able to turn over again, you can still get degradation. And so they're often not as susceptible to point mutation-based resistance. But of course, you can get resistance by other mechanisms, right? And it's already known from the clinic, if you silence expression of the ligase that the molecule needs to degrade, then of course the molecule stops working. So it's not like they're impervious to resistance. It's just the resistance mechanisms are often orthogonal to what you get with a traditional inhibitor. Then this third point is the ability to eliminate the scaffolding function, or sometimes you get a bystander effect where you recruit to protein A, but because protein A is binding to protein B gets degraded. So you can actually sometimes use sort of a nearest buddy effect to get degradation of another protein target. And that relates to the fourth point of, especially in cancer, we have a long list of, highly credentialed potential drug targets that we've never been able to drug. And so this might be an approach for getting at those proteins, which is great for chemical biology because we all appreciate how difficult it is to find targets and to validate them. And really that last step of finding a druggable approach is really rate limiting for a lot of projects. And then finally, like covalent inhibitors, you just need enough exposure of the drug to induce degradation. And then the pharmacology is driven by degradation. You don't need to have the drug there 24/7. And actually, ideally, the PK of the degrader compound should be quite short because, once the molecule is finished degrading the catalytically, most efficient complexes, it goes after things later and later. And so everything has a kinetics for getting degraded. And so the ideal molecule, for example, might have a liability in the linker. So then after a couple hours of exposure, the molecule degrades and stops being a degrader of other protein targets. So what does this look like from a structural perspective? And so Roddick in Eric's lab was able to get a crystal structure of BRD4 bound to cereblon and DDB1. And as we expected, the molecule is like this little noodle here sitting between cereblon and BRD4, inducing this large protein-protein interface. And what we observed by looking at multiple different classes of molecules is that different small molecules induced different orientations of the protein-protein interaction. So basically the small molecule is dictating how BRD4 docks onto cereblon. So here's two molecules, DBET57 and DBET23. And so that's really exciting because it means that the protein-protein interaction surfaces are going to be different, differently induced by these two small molecules. And when we saw that result, we started thinking we might be able to get selectivity for different bromodomain containing proteins. And so there, in our genome, we've got BRD2, 3, 4, and T. And the acetyllysine binding pockets of these four proteins are very, very similar. So what Zhisheng in the lab was able to do was try to look at, is there a unique protein-protein conformation that's uniquely enabled for one of the compounds? And the way we do this is we use this nice Rosetta docking software, and it can predict, the various docking poses. Then you can look at these structures and get an idea of the disposition of the ligand that binds to bromodomain, the ligand that binds to cereblon, what kind of a length of linker you might need, how rigid, what's the trajectory of the angles. Of course, you can't make a precise prediction, but you can get a general idea. And then through synthesis of compounds and testing, you can find ones that enable unique conformations. And so he was able to make this compound 2,147 that turned out to be a unique degrader of BRD4 and then not degrade BRD2 and 3, because the way it tries to dimerize 2 and 3 is not as conducive as the way it dimerizes BRD4. So the selectivity here is not from the small molecule protein interaction, it's from the protein-protein interface. So that was really exciting. And I'll show in a moment that this is the principle behind why we get really selective degraders for kinases. The other technology that we've frequently implemented for these degraders is quantitative proteomics. So this is a really powerful approach using this TMT labeling, where you can quantify several thousand up to 6,000 or 7,000 proteins in a single run, and then really ask on a proteome level, how selective is your degrader molecule? And this is an example of a very selective compound. You can see here on the y-axis is the log fold change for BRD4, and then a p-value along the x-axis. And you'll see various profiles. So this is an example of a selective compound. You can also have compounds that will degrade, up to 50 proteins. So if we're able to get selectivity for BRD4, now what we're trying to work on is a further level of selectivity, is to get selectivity for specific translocations. And actually the original reason that Jay's lab started working on BRD4 is there's an oncogenic fusion that forms in midline carcinoma called BRD4 NUT. So midline carcinoma is a really horrible, aggressive lung tumor. And what we were able to show here is that the BRD4 selective degrader could degrade the fusion protein, as could the pan degrader DBET6. But what we haven't been able to achieve yet is a degrader only of the fusion protein and not of wild type BRD4. The problem with wild type BRD4 degradation is that's a very powerful general transcriptional block associated with quite a bit of cytotoxicity. So what we really like to do is only degrade the cancer causing fusion protein. And so what we're trying to do is find a ligase that we can recruit that cares about the nut fusion part of the protein, so it can recognize that that fusion protein is there, and we want to eliminate it. All right, let me pivot now and talk about our efforts doing protein kinase degraders. So I think everyone in this audience is familiar with the kinome, given the folks that live and work at UCSD. Two major challenges in the field have been drug resistance. So even though there are many great examples of efficacious kinase drugs, patients ultimately become resistant. And then the other problem has been a lack of generality of finding single oncogenic drivers of very complex tumor types. But we thought that if we could efficiently make kinase degraders, we might be able to go after a larger set of kinases, either because we need the better selectivity or because of cases like HER3, where the kinase is a pseudokinase, primarily serving a scaffolding function that we'd be able to eliminate with a degrader. And we actually tried initially with HER3 because we thought that was a clear target. And after making about 80 compounds targeting HER3, we utterly failed. So we had to take a step back and try to figure out, why did we fail with HER3? And so instead, what we tried to do is make intentionally promiscuous degraders. So here the idea was, let's find a compound and a series of compounds that bind kinases promiscuously and figure out how many can be degraded. And so the first lesson that we saw was that it was actually quite difficult to make a really promiscuous degrader. So we thought, like with this compound, based on a desmethoxy TAE 684 compound, this compound can bind several hundred kinases. But when you turn it into a degrader, it only degrades 20 to 30 kinases. And we got excited about that because then we could maybe optimize and make degraders of those kinases, but we really didn't know how many other kinases would be able to be degraded. And actually, this was a project that Fleur headed up in the lab together with Catherine Donovan in Eric's lab, was to really try to systematically look across the kinome and find more kinases that could be degraded. So using sort of a team science approach, we accumulated a lot of different kinase degraders and then put them through a proteomics workflow where we would treat cells and then after 5 hours, look at how the proteome was affected. And just some high level, take-home messages from this story. We actually made this resource available through a website, so you can search for your favorite kinase and see if we have a degrader of it. One thing that stood out was sometimes quite promiscuous binding compounds like this compound based on dasatinib turn into be a very selective inhibitor of a particular kinase. So this compound, despite CSK being perhaps number 10 or 15 on the list in terms of binding affinity, it was the most readily degraded kinase. So promiscuous binder leads to a selective degrader. And we spent a lot of time, figuring out, the reasons for that. And, on one level, it's sort of the expected conclusion, right? To be an effective degrader, you got to bind the target, you got to dimerize, you got to ubiquitolate. Ubiquitylation has to lead to degradation. So many links along that chain have to happen, correctly. And we, then went in and tried to figure out what are the rules that govern that that selectivity. This is a little bit of a complicated slide here, but I'm just showing on the right four compounds where we quantified whether we could detect a target in degradation or whether we could detect target engagement. And so what you can see is, depending on the compound, different sets of the targets categorized either having no target engagement, as measured by a pull down assay versus whether they were degraded. And so the way I think about this is that some molecules are actually not very good catalysts. And in order to work, they got to have good affinity, and you got to sit there for a long time for the ubiquitination to happen. And then there's other ones where the way they recruit is catalytically, very efficient. And despite, really wimpy binding, you can still get, catalysis. And so anybody who's familiar with, making small molecule organocatalysts will readily understand this phenomena. It's very hard to figure out how to stabilize the transition state. There's lots of magic, that happens there in terms of molecular dynamics and conformations that, can't easily be, predicted. And so that's why this empirical approach of doing the mass spectrometry is really key to find these low affinity interactions that still lead to very efficient degradation. We also try to compare the efficiency of recruiting different kinds of ligases. So about 90% of the work that's been done in the field has been with two ligases, Cerebellum and VHL. So we made a series of matched compounds and then asked, when do you see preferential degradation with a VHL-based recruiter versus a Cerebellum-based recruiter? In general, you can see pretty good congruence. Many targets are well degraded by both, but then you can see some, that are better degraded by Cerebellum-based recruiter and some based on VHL. And then again, that makes sense, right? You're fundamentally forming different protein-protein complexes, and the degree of molecular complementarity can be different for different ligases. And this is going to be a really fruitful area for future research because there are, so many other ligases. You're probably going to be able to find ones that are either uniquely expressed to get, tissue-specific degradation, or have a unique interface that allow you to get, a high degree of selectivity for a given target. So once you have that big database of information, how do you go from the dynamite, version of selectivity to a compound that's a really selective degrader? And I'll just show you how we did that for one target of interest called Burton's tyrosine kinase. So this is a validated, clinically established kinase target in a variety of B-cell tumors and chronic lymphocytic leukemia, Waldenstrom's, macroglobulinemia. And there's a blockbuster drug that's been developed called ibrutinib. And so ibrutinib, the way it works is it blocks the ATP binding site and it forms a covalent bond to cysteine 4D1 and inactivates the kinase. Unfortunately, after you've been on this drug for a long time, you may eventually evolve a clone that no longer has the cysteine. So this is not a catalytically important cysteine, it just happens to be there. And so if you get that cysteine mutation, then ibrutinib stops working and the tumor can grow again. And so we thought, why don't we try to make a degrader that doesn't require cysteine ligation? And so Dennis Dormanovsky in the lab took a very good BTK binder that had been highly optimized, a very selective ATP competitive BTK binder, and then made a degrader out of it. And whenever we do this, we'll often make a matched pair of compounds where we have the compound that can bind cereblon, and then we deactivate cereblon binding either by methylation of the glutaramide or by removing one of the carbonyl groups. And that's a really important control because these molecules are like Swiss Army knives. They still have the kinase inhibitor function, but then they also have the degrader function. And so you want to be able to ask pharmacologically what pharmacology comes from degradation, what comes from inhibition. And by comparing these matched pair of compounds, you can ask, what is the degradation part of the pharmacology and how is that, contributing? We also have to develop a series of other assays. For example, I mentioned measuring target dimerization by TR-FRET. And then we've got some nice competitive cellular engagement assays where we can measure, how much of the degrader is actually getting into the cell. So this compound worked, really well, 2 hours treatment, 100 nanomolar, totally eliminate BTK. And because this compound is not cysteine directed, it's an equally good degrader of wild type BTK and the cysteine mutation. So that's the good news. You can overcome the cysteine mutation. Of course, as I mentioned before, you can get other types of resistance mechanisms to this compound, including mutations that affect the ability to dimerize. So instead of getting a mutation in the drug binding site, you get a mutation at the protein-protein interface with the ligase. So you can select for that. The other thing is because these molecules are quite big, you can often get overexpression of an ABC or other transporter protein that pumps the PROTAC out of the cancer cell. But despite being a very primitive tool compound, I think we only made about 10 or 15 compounds for this particular effort. We were able to get a compound that worked well in vivo. So in collaboration with David Weinstock's lab, able to show activity in a mantle cell lymphoma model. And the great thing about degraders is you don't have to spend a lot of time finding a biomarker. You can just look for the target degradation by Western blotting or by flow cytometry and corroborate that you're degrading your target of interest. And you can see, even with this primitive tool compound, we could get a really dramatic extension of survival compared to ibrutinib. So I've got no doubt that somebody could come along and make a really potent BTK degrader, and probably that'll be tried clinically soon. So we think this sort of non-specific ligand degrader approach is a great way to go in and establish if a given gene family is targetable, whether it be a hydrolase or an ion channel or other, and then figure out what subset of that family might be more easily addressed. Because clearly, as you can see with the kinases, from our effort, we found, maybe 150 kinases or so easily degraded, and probably with more effort, more will be easily degraded. But there's a huge difference in how challenging it is to optimize a difficult to degrade kinase versus an easy to degrade kinase. So establishing the tractability across the target class, I think, is really a useful exercise before, becoming hell-bent on focusing on degrading, one particular protein. What about other targets? So obviously degraders could be interesting in areas outside of cancer. And in some work we did with Priscilla's lab, we did initial proof of concept for hepatitis C virus, where we took an established protease inhibitor called telaprevir, which was moved into the clinic, and it never became an established drug because you get resistance quite quickly to it, and it ended up not having as good activity as some of the polymerase inhibitors. But it was a very good reversible covalent protease inhibitor. And Guangyang in the lab made a degrader, Cerebellum-based degrader compound off of it. And again, we make this negative control with this methylation at the glutaramide. And you can see you get very nice degradation of the protease. And just like we saw with BTK and some of the kinase targets, it's much less susceptible to the typical point mutation-based resistance. So here you see with telaprevir, these different protease mutations really changing the EC50 quite substantially. These same mutations do not affect the the degrader. Again, because a small change in drug occupancy doesn't fundamentally alter the fact that the compound can be degrading to an effective level. So this is just a proof of concept because here you could find a orthosteric traditional inhibitor, but viruses have all sorts of other proteins that are these sort of Houdini type proteins that have many functions, and if we're able to degrade those proteins, I think those are going to be really interesting targets. And so we're working on several of those now with Priscilla's lab and trying to show that some of them can be drugs. So I think that's pretty promising. And again, it makes a lot of sense because viruses use this exact strategy on the host cell all the time, right? So most viruses have some way to inactivate the innate immune system through degradation of the sting receptor or RIG-I or other types of innate immune sensors. So it actually makes a lot of sense that you could use the same strategy in reciprocal against viruses. In some really nice work that Fleur led in collaboration with Steve Haggerty's lab, we looked at tau. And so here the idea was just to take these PET imaging probes and try to see if they could be turned into degraders. And so other chemists had found these neat compounds which seemed to be able to bind different forms of tau, and they'd made these F19 PET imaging agents. We actually didn't have any idea of exactly how they bound and what structures they recognized, but amazingly, when these compounds were made and tested, they really seemed to be able to eliminate tau, specifically mutant forms of tau in these IPS-induced neuronal cultures. And because the compounds themselves have some mutant selectivity, And also probably because of the protein homeostasis is quite different when you have mutant tau versus healthy tau. They actually seem to be somewhat mutant selective. So you can see here, we're not eliminating tau in these healthy control neurons. I mean, a lot of work needs to be done to further corroborate this, but I definitely think the idea that degraders can be useful for protein aggregation, based diseases is really promising. And so Fleur is going to figure that out in the next 5 years. So in the last couple of minutes here, actually lost, how much time do I have, Fleur? Another 10 minutes, great. So that's perfect amount of time. So let me quickly talk about this DTAG system. So here the idea was, if we don't have a ligand for your target of interest and you don't want to go through a whole chemistry campaign, why don't we just develop a nice degradation tag? And people had done this before, but many of the compounds that were used were not that convenient and readily accessible. And so Benamnabet designed this system where we took mutant FKBP, which is a favorite protein of chemical biologists because there's some very good small molecule ligands for it, and created fusion proteins with that ligand. And then we developed a whole library of degrader molecules, and we found both cerebellum and VHL-based degrader molecules that could induce ubiquitination of the tag. And because the tag is fused to the target of interest, you get complete degradation. And so this was really exciting because unlike RNAi-based approaches, which take at least 6 to 12 hours to really induce, here you could get really rapid degradation, often in less than an hour. And so if you wanted to look at fast biology, I mean, not super fast biology, but intermediately fast biology, you might be able to do it with this degrader system. And then obviously because of the CRISPR-Cas9 revolution, the ability to, introduce this construct, in endogenously and do knock-ins. Actually, I just saw a paper published last month where people have now made a transgenic mouse with a DTAG version of a protein using CRISPR-Cas9 in embryos, and then making it a transgenic mouse with this fusion, and then you can degrade. And the molecules we made actually can be used in mice as well. So just a couple of examples here of people using this approach. So here, before we were able to make small molecules that were selective for BRD4, we used the DTAG system to specifically degrade BRD4. And what you see here is you get a much milder effect on proliferation if you only degrade BRD4 versus you have a pan degrader of all the bromodomains. And if you look in the literature, you see hundreds of papers, with JQ1 saying it's BRD4, but it's actually a pan bromodomain inhibition. And what you might see from BRD4 could actually be a lot more selective. A nice paper from Rick Young's lab took a transcription factor that has absolutely no ligands, YY1, and really established, what happens when you eliminate YY1 from mouse ES cells, and looking at the transcriptional consequences, or validating putative oncogenic drivers. A nice paper from Brunetti et al. Degrading NPM1C. So this has become a really useful system, and we got so many requests that we ended up making sort of an online system where you can get the vectors now from Adgene, and now there's several vendors that sell the compound. So I think that's going to be really useful. And people are now evolving other tags as well. So there's a nice paper just published with a mutant form of BRD4 from the Alessio group. So you'll be able to, for example, introduce 2 different tags on 2 different proteins and have 2 different small molecules that can control degradation of protein A and protein B that allow you to do interesting combinatorial experiments. So I think the age of small molecule degradable tags is really dawning, and it's going to be quite convenient. This is not, it's a good system for eliminating the function. It's not a good system for establishing how easy it's going to be to degrade your protein, because the ubiquitination here happens on the tag, not on the endogenous protein. So sometimes you then, you validate the target, but then when you go and try to make a degrader of the wild type protein, that can end up being a really big lift. Okay, so the molecules I talked about so far have primarily been these bivalent PROTACs, and there's a huge space here remaining for optimization. As I mentioned, there's about 800 different ligases. We've only got, as a field, successful recruitment of about 10 different ligases, with 90% of the work being on Cerebellum and VHL. So a lot of work now figuring out how do you find new small molecules that recognize ligases, and then which ones can be co-opted into a degrader molecule. But people are also focused on the idea of these molecular glues, right? So here's the crystal structure of Cerebellum with a segment of IKZF1, which is, if you remember back from the beginning, is the target that's degraded in myeloma. And what you see here is, yes, there's molecular interaction, between the small molecule and Cerebellum and between IKZF1, but fundamentally it's a softball in a softball mitt type interaction. Most of the interaction is protein-protein interface. And so really what you're trying to do is, cause the softball mitt to be able to grab, balls of different sizes and different textures. And what are the rules, behind doing that? And so we and a number of other labs have been systematically exploring the thalidomide structure and asking, how can you dial in and out different substrates? It turns out that most of the substrates have this so-called G loop. It looks like this, that binds here, and there's different molecular interactions, that form, but there's not enough sequence conservation in these loops to find them informatically. You've got to find them experimentally. And so here are some examples of some different neosubstrates that have been established. So IKZF1 and 3, which are efficacy driving targets. There's a kinase, casein kinase 1, that has the loop that can be degraded, and that's an important target in a certain type of MDS syndrome. There's SAL4, which is the anti-target we try to avoid. There's a beautiful series of compounds developed by Celgene here just up the road that are degraders of a translation factor called GSPT1. And then there's been some work on another zinc finger transcription factor called ZFP91. So we built out a library of several hundred of these IMID compounds using combinatorial chemistry. And we were also able to screen and find some very good GSPT1 degraders. And when you inhibit this translation termination factor, you get a cytotoxic effect in a variety of leukemic cell lines. And we've taken that library now and found a whole bunch of other zinc finger targets. We've also explored another scaffold that was initially discovered by a group in Japan at Asai called Indusulum. So Indusulum binds to a different E3 ligase. Instead of binding to Cerebellum, it binds to decaf15. And we built out a big library of this type of compound, hoping we would be able to degrade other proteins. And so the proteins that Indusulum degrades, there's a splicing factor called RBM39. Unfortunately, despite making a quite substantial library, we were only able to degrade one other protein called RBM23. And when we looked at why that was, it turns out that the domain that they recognized, the RRM domain, is the most conserved between RBM39 and RBM23. And then a whole beautiful series of studies done by our collaborator, Eric Fisher's lab, they were able to figure out the reason that was much more difficult to get general targeting from this ligase is that the molecular recognition with the RRM domain is over a much larger protein-protein interface. So it was much harder to fundamentally change the recognition with a small molecule, versus in the case of Cerebellum, it's a much more of a glancing protein-protein interaction, and that can be reprogrammed quite a bit more than this interaction. So that's part of it. It's probably also related to molecular dynamics and how much conformational heterogeneity you can get through introduction of a small molecule. But I just show you here. So here's the decaf RBM interaction, really programmed biologically with that protein-protein interface. Whereas here's the interaction between Cerebellum and one of the neosubstrates. So for this glue approach, we think there's going to be a smaller subset of ligases where the plasticity and the degree of difference you can get through the small molecule is going to be greater, and fundamentally those will be easier to reprogram and get them to degrade other targets. So when we think about this in sort of a forward sense, we're doing work both on the bivalent, PROTACs, which have the advantage that you can exploit known ligands and their sort of systematic way of going about making them, versus these glue molecules, which are potentially more exciting, mainly because they often encourage you to work on things that you weren't working on before because there wasn't a ligand. So things like IKZF1 and 3, any chemist would have told you these were undruggable, but it turns out by this degradation mechanism, they're highly druggable. So by not looking for your keys under the lamppost with these glues, you often get into some very interesting pharmacology. So where is this field headed? I think really this is just a subset of what's going to become a much larger field around what we're calling, and others are calling, proximity-based therapeutics, right? So these are compounds that fundamentally get their pharmacology not because of the small molecule being an inhibitor, but because the small molecule can hijack an endogenous biochemical event. And right, so far everything I've been talking about has been hijacking some E3 ligases, but you can imagine hijacking all sorts of other intracellular protein functions, right? And there's been some preliminary work from the Broad targeting kinases. So instead of making a kinase inhibitor, they're making kinase recruiters to induce specific, phosphorylation events. You can imagine, in the transcription field, small molecules that move transcription factors around or that change nuclear cytoplasmic localization. Those might also have very interesting, pharmacology. So the idea here is really thinking of, molecular glues that can reprogram functions that are already present, inside of cells. And when you think about it, kinases, to do that to some degree, they transfer, phosphate to another protein to induce neofunction to that protein, and they do that through the recognition of the kinase of the substrate, right? It's not, ATP provides the phosphate, but the protein-protein interface, provides the selectivity. So really, to get these really selective compounds, we've got to, marry the world of small molecule recruitment with the enzymatic functions that we want to reprogram inside of cells. And I think that's going to have a really exciting effect on the types of proteins that we're able to modulate in the future. So let me stop here and thank some really fantastic people that led this project. All the work that we do in the degrader space has been in close collaboration with Eric Fisher's lab, who is upstairs from us at Dana-Farber, and two really talented scientists in his lab led a lot of the sub-teams, Radek on the structural biology and Catherine in the proteomics realm. Binam was the brains behind the DTAG system. Ting Hu is a senior chemist in the lab who enabled a lot of the chemistry design. And as I mentioned, Fleur led our effort on the degradable kinome and on the tau degraders. We had really great collaborations with a number of other labs, and I'll stop there and take any questions. Yeah, so a number of companies, so the question was, how bad are the drug-like properties for these bivalent compounds? So I know of at least 8 or 9, clinical candidates that are going forward. The major challenge has been around, so the reason that they can work at all is that instead of needing to work at micromolar levels, they work at nanomolar levels. So bioavailability, cell penetration, those things are compensated for by the increased potency. What's not compensated for is the biophysic limitations of the compounds, drug solubility, stability, formulation. So if I were to put a number on it, I'd say it's, 3 or 4 times as hard to get a development candidate from a bivalent degrader than a classically sort of well-behaved small molecule. The other part of it is just learning pains. A lot of organizations are figuring out how to make these. So I think as the rules of the game evolve, it's going to get easier. And then of course on the finance side, the 10 bets that are going in the clinic, if 1 or 2 of those works out well, then everybody will be excited and try harder.
Are they all given as an infusion or any of them?
So no, so people are making orally bioavailable Protacs. There are a couple people that are making ones that are for IV, but they're oral, and there's even, you know, claims that people have gotten reasonable CNS exposure from some of them.
With your extensive experience here, and you talked about cell penetration, if you took all of these degraders with respect to substrates, would you find the most susceptible to be intracellular or extracellular, or proteins that would have both exposures?
Yeah, so there's good examples, obviously, of cytoplasmic proteins being degraded, nuclear and membrane bound. All the degraders I talked about today, you know, act from the intracellular side. There's a beautiful paper from Carolyn Bertozzi's lab with Litax. So there they had a bivalent approach where they had an antibody to the extracellular portion and then a carbohydrate binding moiety that caused binding to an endocytotic receptor and then, you know, going into the lysosome. So there are good examples now of bivalence for extracellular. We've definitely seen examples where one subcompartment of the protein is protected from degradation. We think because it's in a complex that prevents the ligase from getting there and being, you know, being recognized. So some examples where you can get, you know, good degradation cytoplasmically, but then nuclear, you know, degradation is less. So, you know, anything that gets in the way of the ligase being able to be recruited, you know, then you can, you know, not see the degradation. I also, you know, as I said, 90% of the work on Cerebellum and VHL. So as people expand out with other ligases, you know, some of them might be better at getting proteins in other locations.
That was really great. So I wondered, you know, cancer seems simple, kind of, you just want to get rid of the bad protein. What about neurodegenerative diseases where you don't want to just destroy the cell, you want to just have a very selective, a one mutant compared to the wild type?
Yeah, so I think if the mutation, you know, either affects, you know, recruitment to the ligase, or there's a fundamental difference in the protein homeostasis, that can lead to selectivity of the compound. The way we've sort of looked at that has been pretty crude. But for example, when we do proteomics and you change protein homeostasis, for example, by putting in a deubiquitinating inhibitor, you fundamentally change, you know, what gets degraded. So it's clearly a dynamic balance between the intrinsic, you know, protein homeostasis and then what the ligand does. But people are looking in other directions. So there's a beautiful paper from Dan Nomura's lab. They've made dubtacs. So they made recruiters of deubiquitinating enzymes to actually stabilize proteins, which you might want to do, you know, in other settings. So I think there's going to be, you know, appropriately balanced mechanisms where you might actually find that the ligase expression is different in the disease state, and then that becomes the basis of the selectivity. It's not because of degradation, it's because of the ligase homeostasis.
That's a really great talk, actually. I have a question, actually. You probably know that thalidomide actually was initially proposed by Judah Folkman's group, you know, to be an inhibitor of angiogenesis. They claimed that, you know, the mechanism of, you know, tumor growth inhibition was mediated by that. But in light of the things that you described today at the work, do you think there is any evidence that this is true? Because this was always a little bit of a controversial view. What is there any evidence that, you know, actually blocks angiogenesis, or an entirely different mechanism, as you said, you know, by ubiquity, by degradation, actually?
Yeah, I mean, there are some people working on alternate, you know, potential, you know, mechanisms, at least in the cellular systems that we've looked at in myeloma cells and leukemic cells. You know, if you get rid of cerebellum, you know, you pretty much lose all the pharmacology of thalidomide. So I'm pretty convinced that that's the majority of the story in those settings. You know, there's obviously thalidomide gets metabolized to all sorts of different things in vivo, and possibly, you know, there's additional mechanisms at play. You know, it has very strong anti-angiogenic phenotypes in certain settings, which may derive from from something else. Part of the conundrum is we don't actually know the natural ligands for cereblon, and the binding pocket of thalidomide is a beautifully conserved pocket across many species, and we actually don't know why Mother Nature made that pocket. And we think it might be, we think thalidomide might mimic some kind of a post-translational modification. The glutaramide, actually looks like a cyclization of a glutamic or an aspartic acid onto the C-terminus, but no one's really been able to prove, what the natural degron is. And so I think that's part of the problem to your question, is that because we don't know enough about cereblon biology, it's hard to know if that's going to really be the whole story with thalidomide. But in terms of, what we're doing of just sort of hijacking it for other degradation purposes, the story at least is correct enough for that part of it to work.
Okay, thank you.
Yeah.
Hi, so may I ask a question?
Please.
Yep. So great, thank you so much. It was amazing to listen you here. So I'm a postdoc from University of Minnesota. Actually, you have covered both areas, the cancer and the HCV, and both of our interests. So I have two quick questions, if I could ask that. So what is your success in the HCV inhibitors? So in your slides, you have shown some of the NS3 5A. So are you looking for some other non-structural proteins target for the degradation? And the second thing is regarding the cancer. So you have mostly shared the work on the degrader with the CK1. So do you see that there are more targets in the cyclin-based kinases which we can hit and we can dig into for the degradation? Thank you so much.
Yeah, both good questions. If you plot, the number of degraders with time, we're in an exponential growth phase right now. So people are trying to turn almost every ligand out there into a degrader, and it's impressive, how well it works. I wish people would publish the ones that didn't work. Whenever we try to do that, it's actually twice as hard as publishing something that does work. So it'd be nice. We don't really know what the denominator looks like that, but I think we're going to see many, successful examples. But in terms of, which of those examples can really be made into a super duper, potent degrader, that's only been done on a limited number of cases. So for, BRD4 and some of the kinases, people have been able to make, like picomolar level degraders. So we don't actually know the rules for when you can get to that level of potency. And then in terms of your other question about the the viral factors, I think people are starting to ramp up their efforts in that in that arena, and then for neurodegeneration, and also for even some companies that really only want to pursue it for cancer initially, but I think eventually we'll see it being used for autoimmune and inflammation-based disease targets as well.
Okay, thank you.
If there are no more questions, let's thank Nathanael for an amazing one last. Okay.
Okay. So the question is, have you used unbiased, unperturbed cell lines, no tags, to screen for novel degraders? What would be the readout in that scenario? Have you seen any data on how protein degradation shows up in transcriptomic data? Yeah, so sorry I didn't make that clear, but like the gradable kinome effort was done in a leukemic, unperturbed cell line, just in a a normal cancer cell line. So, and there the readout was quantitative proteomics, and that's by far the best and the most unbiased way at looking at protein degradation, and it's gradually becoming more and more high throughput, and you're able to profile more cell types and more compounds. So that's definitely the way to do that. In that study, we did try to quantify which changes derive from protein based effects versus transcription, And most of them were protein-based effects. In some cases, the loss of the protein then, signals back to transcription, and then you get a transcriptional perturbation as well. And obviously, certain proteins, when you degrade them, like CDK9 and BRD4, very quickly that signals down to transcription. So you definitely also see some transcriptional changes. But these bivalents, really do work at the level of proteins.
Is that it? No less questions online.
I think we're good.
All right, in that case, thank you so much for an amazing talk.